Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 16 from our Genetics Essentials, Concepts and Connections 4th edition textbook. This chapter deals with cancer and cancer genetics. So let's go ahead and get started. Obviously, cancer is a huge problem, a huge issue in the healthcare field. 1 in 5 women and 1 in 4 men will ultimately succumb to cancer, and that will be the reason for their passing. Cancer is not a single disease. Rather, it is a heterogeneous group of disorders characterized by the presence of cells that do not respond to normal control of division. Remember, we discussed the cell cycle, how you have interphase, G1, S, G2, and then there's mitosis, and there are important signals in the cell that direct the cell when to divide, when not to divide. Do you remember? And if a cell does not get the signal to divide, do you remember how the cell could exit the cell cycle between the G1 and S transition? Remember, if there is no proliferative signal, the cell will exit the cell cycle and enter what's known as G0, where the cell is directed to just function as a cell, but not to proliferate, right? And as a multicellular creature like you and me, it's very, very important that our cells know when it's appropriate to divide and when it is not appropriate to divide, right? Because you want a certain layer of skin cells. You want a certain density of liver cells. You want a certain arrangement of lung cells. Does that make sense? You wouldn't want a lung cell to just willy-nilly start dividing over and over and over again, right? That is called a loss of control of the cell cycle. And that could lead to a ball of cells, right? If one cell just kept dividing and dividing and dividing with no control, that would lead to a ball of cells, wouldn't it? And that is known as a tumor, right? That is a tumor. That's the beginning of cancer. And you can see these tumors in tissues. Once the tumors form, you can see these tumors. Again, cancer is cells that divide rapidly and continuously. They're not going to cease. They just keep dividing, creating tumors. Tumors are little balls of cells, right? So you have little clumps of cells. And the problem with tumors is that they crowd out normal cells and eventually rob the healthy tissues of nutrients. Think about it. If I have way too many lung cells and those are taking up space from other cells, well then they're going to rob the nutrients. They're going to not only rob the nutrients. Look at this picture here, figure A. Let me show you figure A. Figure A is a metastatic breast cancer mass in a human liver. So what you're actually seeing is a part of human liver. Here, you're seeing part of the human liver, and these white spots are breast cancer tumors. You have breast cancer cells in the liver, and you might be wondering, why is there breast cancer or breast tissue in the liver? Well, because of a phenomenon known as metastasis. Metastasis is spread of tumors from one location to another. So in this patient, you see this patient with this liver? This patient originally developed breast cancer, so breast tumors. At some point, some of those tumor cells left the breast tissue and traveled either through the bloodstream or the lymphatic system to other organs, right? And then once it reached other organs, it began to grow in those organs. So that's how you can tell, by the way, they do a biopsy of your liver and then you found out you have breast cancer, and you're like, how did you find out I have breast cancer from my liver? That's how. The cancer starts in one location with one cell usually, right? So it would be a cell in your breast tissue that's lost control of the cell cycle and decides it's going to divide. At some point, it spreads from a lump in your breast, it spreads to the liver, it spreads to the lungs, it spreads to the bone marrow, it spreads to the spinal cord, it spreads everywhere. That's called metastasis. And that's how they could figure out what type of cancer you have, even though they took a biopsy from a different tissue. Does that make sense? Now, can you see how that would be a problem here? If you take a look at this liver, can you see how having a bunch of breast tissue taking up a bunch of space in your liver would be a bad thing? Because all of these breast tissue cells, all of these breast cancer cells inside of your liver are eating up resources, oxygen, nutrients, amino acids, sugars, fats from the blood, right? And they're not really contributing anything. In fact, not only are they draining your nutrients, they're also occluding the liver cells and preventing them from functioning correctly. And if we were to section through these liver cells and take a close look with the microscope, you can see with a light micrograph of liver section with tumors, the white cells are tumor cells, the normal cells are here, the darker cells are the liver cells. Notice how the white cells are just intruding on and occluding the healthy cells. This is going to lead to eventually severe illness and death, right? Imagine all these tumors all over your body sucking up resources and preventing the function of your normal organs. That's going to lead to death. And speaking of that, look, here's a rundown of the new cases. This was for 2017. New cases of each type of cancer and the number of deaths caused. So, for example, in 2017, 255,000 breast cancer cases were diagnosed, and that led to 41,000 deaths. So we could do a quick calculation here. 255,000. Hold on, let's do the other way, of course. 41,000 deaths. 41,000 deaths. 255,000 cases. That's 16% of people with breast cancer passed away. So this is horrible. The cases are still high, and the number of deaths are still high. Medicine has not found a way to cure all cancers. Now, there are techniques that we have, like radiation therapy, chemotherapy, and a number of different techniques we have, and anti-VEGF treatments, which stops angiogenesis. We have a number of different drugs and techniques. Bone marrow transplants, for instance, for leukemia and lymphoma. But we're still not to the place yet of being able to treat all cancers. In fact, some cancers are almost 100% lethal. So, for instance, look at this one. Pancreatic cancer is a horrible type of cancer to have. Let me bring up my calculator again. With pancreatic cancer, we're talking 43,000 deaths in 53,000 cases. That's an 81% chance of death, right? So high mortality. Some cancers have a very high mortality. 81%, for instance, is a high mortality risk rate. And some cancers have a lower mortality risk. So let's look at, one of the lower ones would be melanoma. 9,700 cases, 9,700 deaths in 87,000 cases. So only an 11%. Now, that's still high, don't get me wrong. Melanoma deaths are still high, but 11%. But with pancreatic cancer, you saw it was 80%. And you might be wondering why. Well, a lot of times with cancer, it all has to do with when you catch it, with like when do you discover the tumors? Do you discover them early on or too late? How aggressive is the cancer? And with melanoma, luckily, it's on the surface of your skin. And a lot of people nowadays go to dermatologists, or they know what they're looking for. They're looking for irregular shaped things that look like moles. So you catch them more often, right? That would lead to better outcomes. You catch the cancer earlier, better outcomes. This is why they recommend mammograms, so you catch lumps in the breast early. They recommend dermatology visits, so you catch weird growths early. They recommend colonoscopies, so you catch colon cancer early, the little polyps early. But with your pancreas, think about your pancreas. I mean, how are you gonna, you don't even know what you're looking for, or, a lot of times by the time you've figured out there's something wrong with the pancreas, it's already kind of too late. It's already kind of metastasized, right? Does that make sense? Now, there's, of course, more to it than just that, but one of the biggest things you can do is to catch these things early, because we're going to learn in a minute that there's a progression in cancer. It's not just cancer happens and then it spreads immediately and then it's bad outcomes. A cancer cell needs to incur a series of mutations and gain a series of abilities before it's able to spread and cause a huge issue. So this is the process of tumor formation we're talking about. So normal cells grow, divide, and mature and die in response to internal and external signals, right? Your skin cell knows when it's appropriate to divide, grow, stop dividing, start dividing. Does that make sense? And it's based on signaling. There are growth factor signals. There are endocrine signals. There are paracrine signals. There are all of these signaling molecules that speak to your skin cells, and also there are signals inside of your skin cells that regulate cell cycle in the skin cell. So your skin cells should know, okay, I shouldn't be dividing right now. I've got the signal to divide. Now, let's do all the internal checks so that we could start the process of dividing. Oh, the DNA's damaged? Okay, my internal signals are telling me the DNA is damaged, so I'm not gonna divide. Is there a problem with metaphase? Is there a problem with the sister chromatids lining up the center of the cell? Well, then let's not divide, right? Does that make sense? You have external signals. Most of these are growth factors, and you've got internal signals. Most of these are checkpoints. If you've heard of the checkpoint proteins during the cell cycle, all of these have to work in concert in order for your cells to direct normal cell cycle, normal growth, right? So cancer cells, they have disrupted cell cycle signaling. Something is wrong. They're not responding to these internal signals or these external signals correctly anymore. Does that make sense? That causes them to proliferate at an abnormally high rate, forming a tumor. Something went wrong with the cell cycle control. The cell, instead of waiting between G1 and S of interface, it just proceeds into S. Remember that G1S checkpoint of interface? Well, if that checkpoint's gone, the cell will just enter S. It'll go straight from G1 into S. It'll never exit the cell cycle into G0, and it will never stop dividing. And that's what cancer is. You have disrupted control of the cell cycle. And that will form a tumor. A benign tumor is a tumor remains localized. So when they say a benign tumor, a tumor formed, but it didn't progress past that. It didn't metastasize. I remember what metastasis means. Metastasize, metastasis. The tumor cells induce secondary tumors, which means they spread to other locations and start a new tumor. So if your lung cancer metastasizes, it might travel to the liver and grow in the liver. You see, that's metastasis. If it stays as one little tumor in your lung and it never spread, that's a benign tumor, right? But you have to be careful because a benign tumor could incur another mutation and become metastatic. What is a malignant tumor? A malignant tumor is basically one that is metastatic. It's a tumor that invades other tissues. A tumor that invades other tissues. So let's say you have melanoma on your skin, it gets really bad and it starts invading the underlying muscle tissue, the underlying blood vessels. It spreads through the blood to other organs, and that's how melanoma ends up killing, right? Melanoma ends up killing because that spot on your skin invaded, it was malignant, it invaded the other tissues, ultimately going systemic, traveling through the blood or lymph to other spots and growing and taking over, say, your liver or your lungs, etc. So here's the thing that you need to understand, and you might be wondering, why are we discussing so much cancer now in a genetics class? Well, here's why. This is the most appropriate place to discuss cancer because most, if not all, cancers arrive from defects in the DNA. The DNA, you disrupt the DNA, you mutate DNA, that's a surefire way to end up with cancer. Most cancers are not the result of just one mutation, but rather multiple mutations. That's something people don't understand. One thing real quick I want you guys to start thinking about as, biology majors and healthcare professionals. I want you to start thinking about this. Do you think, let me ask you my own concept check, right? This is Dr. D's concept check. Do you think that cancer would be caused by just any mutation in the gene or in a cell, any mutation? Think about that. It says right here, most if not all cancers arrive from defects in the DNA. Basically, that means mutations of some kind. Do you think just any mutation will give you cancer? Nah. What if a cell in my pancreas, right? What if a cell in my pancreas incurred a mutation in the insulin gene, right? Do you think that would lead to cancer in that cell? No, it might lead to a faulty insulin protein, right? For that one cell, but then there's other adjacent cells making perfectly good insulin, so no big harm, no big foul, right? Does that make sense? But what if that same pancreatic cell, what if it incurred a mutation of a gene which encodes for an important regulatory protein, a protein that's job is in the cell cycle? Does that make sense? Now that's a big deal. There are proteins whose job in the cell, their whole reason for being is to take care of the cell cycle. Tell the cell to stop dividing. Tell the cell, okay, divide. Does that make sense? These are your cell cycle control proteins, okay? And we're going to talk about them in a little bit. They're called oncogenes and tumor-suppressor genes. You may have heard of these terms before. Oncogenes and tumor-suppressor genes are genes that code for proteins that have some kind of role in the cell cycle. Does that make sense? And if you mutate those genes, well, then you're going to end up with proteins that don't function correctly, maybe. And if their whole function was to direct the cell cycle, well, then now you're probably going to get cancer. So, you have 25,000 different genes in your human body. Mutating just any gene is not going to give you cancer. But mutating a tumor suppressor gene or an oncogene, these are the genes responsible for the checkpoint proteins, the proteins that tell the cell when to proliferate or when not to proliferate. You see, mutating those genes is a problem. Does that make sense? That's the way I put it. Another analogy is there's a lot of buttons in your car. When you sit in your car, there's all kinds of buttons, but only certain buttons have to do with your radio. Does that make sense? So if I were to start pulling buttons out of your dashboard in your car, one of them might be the hazard light, one of them might be the air conditioning, but only certain ones have to do with the radio. And only the ones with the radio are going to affect radio function. Does that make sense? So same thing with your cells. Only certain genes are going to cause cancer, and those are the genes that play a role in the cell cycle. What else? And again, look what it says here. Most cancers are not caused by one mutation. You need more than one mutation. So it's very rarely, oh, I damaged one gene and now I have cancer. If you damage one gene, that makes you prone to cancer, but then it usually takes a second or a third hit to that cell cycle system. Why? Because there's usually some kind of redundancy. Now, clonal evolution. These are tumor cells acquire more and more mutations, and that's what allows them to become increasingly more aggressive in their proliferative properties, right? So a tumor cell doesn't start out aggressive. A cell becomes a tumor cell when it incurs one or two mutations. Then it gets worse and worse as more mutations accumulate. Does that make sense? I'm going to show you this in figure 16.4. Mutated genes can be inherited, heritable cancer. If there is a mutation and that mutation is germline, meaning it's in the germ cells, remember the spermatogonia, the oocytes, and the sperm or the egg contain those mutant checkpoint genes, then you could be prone to cancer. That's how you can pass on a high likelihood of a cancer. That's why cancers can run in the family, because if you inherit an already mutated gene, and that gene is important in the cell cycle, then you are more prone to that kind of cancer. Does that make sense? However, if I were to go out in the sun and get melanoma one day, I don't have a higher likelihood of passing that on to my child because it's not germline. It didn't affect my sperm or egg. It affected my skin cell. I went out in the sun, and the sun affected and caused those thymine dimers in my skin cell, and then my skin cell developed cancer, right? Melanoma. That would cause cancer in me, but not necessarily, I can't pass that cancer on to my child. The only way for me to pass propensity for, heritable cancer to my child is to have mutations that are germline, that are in the actual sperm or the eggs, right? Does that make sense? All right. So let's carry on with this. Chromosomal abnormalities and carcinogens can also cause cancer. We're going to talk about these things later on. So here you can see this, what is called clonal evolution. Through clonal evolution, tumor cells acquire multiple mutations that allow them to become increasingly aggressive and proliferate. So here's a healthy, happy, normal cell at the top. Then that cell divides, but one of those cells incurs its first mutation. Somehow there's a mutation of an important cell cycle regulatory gene, right? So it says, number one, a cell is predisposed to proliferate at an abnormally high rate. So this did not cause, notice how the first mutation did not cause cancer. This is not a cancerous gene. It's just one that's incurred its first blow, its first mutation, and now is at risk of becoming a cancerous gene, a cancerous cell. So then this cell divides, and now you have two predisposed cells. And number two, a second mutation now occurs, causing the cell to divide even more rapidly. Oh gosh. So now the cell is still not a cancer, but it is growing a little too fast, right? So this may cause irregularities in the tissue. So then you have another cell division. A third mutation happens. Now the cell undergoes structural changes. This is not good. This is probably like a full-blown tumor at this point. These are cells that are just growing and growing and growing and changing shape and not looking normal. Now the cell divides again. There's a fourth mutation. So 4 different genes have now been mutated, 4 different important cell cycle genes. And now the combination of these 4 genes is too much. Now it's a malignant cell. A fourth mutation causes the cell to divide uncontrollably and invade other tissues. So now this would be really bad cancer, right? But it wasn't caused immediately. It wasn't caused by a single mutation. You probably could have caught the tumor here or even here, in the progression. And this is why it's important to constantly have cancer screenings, and catch cancers early, as it's a small lump in the breast or a small mole-like structure on the skin or a small polyp in the colon. That's when the not enough mutations have accumulated. The cancer is not so aggressive. It does not have the ability to metastasize, you see. But once that happens and the metastasis is going on, the cell is at this point, then it becomes increasingly more and more difficult to treat the cancer. Does that make sense? So I also told you about carcinogens a little earlier. The role of environmental factors is huge in cancer because not all cancers are inherited. In fact, most cancers are not inherited. Most cancers are not something you caught from your parents, that was passed down in a gene from your parents. Most cancers are something that your environment had something to do with, right? Like you went out in the sun way too much, right? And that's why you got the melanoma. It wasn't because you inherited a defective gene necessarily from your parents. Many are influenced by environmental factors. Migrant populations typically take on the cancer incidence of their host country. So this is where they showed it's not a genetics thing or a population or species thing. It's an environment thing. Because if someone from one country travels to another country to live, they will adopt those cancer risks of the new country. Does that make sense? So if someone from the Middle East comes to the United States, they're going to start becoming more at risk of the cancers that are here in the United States, and vice versa. If you move to another country, you will be more at risk for the cancers that are more prevalent in that country, showing how important the environment is for cancer risk. Okay, factors contributing to cancer include, but of course are not limited to, tobacco use, obesity, alcohol, UV radiation, X-ray radiation, etc. Do you remember why these things cause cancer? Remember, anything that can affect your gene structure and cause mutations, cause chromosomal breaks, cause chromosome rearrangements or issues, cause anything to do with the DNA, anything that intercalates into the DNA, anything that breaks the DNA or modifies the DNA, anything that looks the wrong way at DNA has the possibility of giving you cancer. This is why it seems like, I'm sure you've heard the expression before, it seems like everything causes cancer. Just throw your hands up in the air and say, oh, everything causes cancer. Well, that may seem to be true, but it's not. They have one thing in common. Tobacco has something in common with obesity, has something in common with alcohol, has something in common with UV, and actually Viruses, did you know, we're going to talk about this at the end of the chapter, but did you know viruses can cause cancer too? Viruses. You're like, a virus cause cancer? Why? Well, think about it. We're going to talk about this at the end of the chapter, but a virus can actually inject its chromosomes or its genome into your cell. And depending on what that genome does, what if, some viruses, they insert their genome into your genome? Well, wouldn't that affect some of your genes? Does that make sense? They can affect your cells in a number of ways by affecting your DNA. Does that make sense? So viruses can mutate or change your DNA. UV can change your DNA, causing those thymine dimers, causing mutations. X-rays can break your DNA, cause double-strand breaks in your DNA. Tobacco chemicals in tobacco called carcinogens, they change your DNA. In fact, carcinogens in general change your DNA. So not everything causes cancer. Anything that can mutate, modify, affect DNA can cause cancer. So that's why it seems like so many disconjointed things cause cancer, because they do have one thing in common. They affect your DNA. And again, real quick, Sean's or Dr. D's concept check. Does affecting any gene cause cancer? No, just the genes that are responsible for the cell cycle. Does that make sense? Remember, genes have different jobs. The insulin gene has the job of regulating blood sugar. Mutating insulin into oblivion is not going to cause cancer. Does that make sense? You could mutate the insulin gene all day long and you're not going to get cancer. But you mutate a gene that is involved, and its whole job in the world is to tell the cell when to divide or when not to divide. That's how you get cancer. You got it? That's the best part to understand. That's the part that takes you from, a beginning biology student to someone who actually understands how a cell works, is understanding that distinction there. So take a look here. Here are examples of geographic variation and the incidence of cancer. So lip cancer, of all things, lip cancer is the most frequent in Canada. You got 15 cases per 100,000 population. But in Brazil, it's only 1 case per 100,000, 15 times less chance, So if you were to move to Newfoundland, Canada, or I guess they pronounce it Newfoundland, you'd be way more likely to develop lip cancer than you are now. And that's strange. Again, migrant populations take on the inherent risks of the places they move to, their host countries. Right? Whereas if you move to Iowa, you're suddenly at huge risk of colon cancer. Whereas in India, Mumbai, you're at very little risk of colon cancer. Why? Well, it's the environment. What are people eating? What are people breathing? What are people drinking? What are the habits? What are the environmental factors? What's going on in that area, right? So let's talk a little bit about what are the genes that cause cancer. Remember, I told you there's a whole subset of genes, and their job is to regulate the cell cycle. Does that make sense? To tell the cell when to divide and to stop the cell from dividing when it's not time to divide, right? Those are, some are proliferative indicators. Proliferative indicators are like growth factors that tell the cell when to divide, and receptors and stuff. And then some are inhibitory factors. They say, whoa, don't divide, right? The DNA's damaged, don't divide. There's inhibitory factors. Those genes are called oncogenes and tumor-suppressor genes. Oncogenes tend to be proliferative. Oncogenes tend to be proliferative factors, and tumor suppressor genes are inhibitory. Oncogene. So this is an important distinction I want you to know. The difference between an oncogene and a proto-oncogene. Okay, this is something that is very important, that you understand the difference between oncogenes and proto-oncogenes. In fact, I'm going to start with proto-oncogene first, because this is the normal gene. Proto-oncogenes are your normal everyday genes. Okay, I want you to know that these are your genes, your normal everyday genes. Proto-oncogenes are your normal everyday genes that are responsible for basic cellular functions in normal cells. But these ones, when mutated the wrong way, can become oncogenes. Okay, they can become bad. Okay, what is an oncogene? What is an oncogene? A mutated proto-oncogene, a mutated copy of a dominant acting stimulatory gene that causes cancer. So think of it as a mutated proto-oncogene. These promote cell proliferation. Did you follow me? I know it's tough, but so proto-oncogenes are your normal wild-type genes. A lot of them play a role in the cell cycle, like stimulating the cell cycle at the appropriate time. Does that make sense? So they're perfectly good. But if you were to mutate them, they could become oncogenes. Oncogenes are mutated proto-oncogenes that are now just telling the cell to divide all the time, not just when it's appropriate to divide. Does that make sense? So let's say a proto-oncogene is the receptor for a growth factor. Okay, you follow me? I'm giving you an example of a proto-oncogene that is the receptor for a growth factor. When the growth factor attaches to this receptor, the receptor tells the cell to divide. Does that make sense? But what if I mutated this receptor for the growth factor to just always tell the cell to divide? Whether there's a growth factor around or not, this receptor will tell the cell to divide. Does that make sense? That would be an oncogene. An oncogene, I've mutated my proto-oncogene, which had a normal job of only telling the cell to divide when the growth factor is around. I've now mutated that into an oncogene, which tells the cell to divide regardless of what's going on ever. You know, so the cell just constantly has this signal to divide. You guys follow me? So proto-oncogenes are your normal cells, your normal genes that when misregulated or mutated, can cause cancer. Oncogenes are those misregulated or mutated proto-oncogenes that constantly stimulate cell growth and cancer. Tumor suppressor genes are also normal everyday genes of yours. Tumor suppressor genes are your normal everyday genes. But these are the ones that inhibit cancer and are recessive. The job of tumor suppressor genes is basically to be the checkpoints. Do you remember the checkpoints? The G1S checkpoint, the late mitosis checkpoint, the G2M checkpoint. There were checkpoints along the cell cycle, do you recall? Tumor suppressor genes, their job is to say, whoa, 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 don't divide because something's wrong, right? No, don't divide. And that's why they're recessive. They are recessive because if you destroy one allele, there's still the other allele to make proteins that say, whoa, 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 don't divide. Does that make sense? You'd have to destroy both alleles to have no policeman to say, don't divide, right? Whereas with oncogenes, they behave in a dominant fashion because you only need one bad guy saying divide, divide, divide. You don't need two bad guys saying divide, divide, divide. So oncogenes behave in a dominant fashion. Tumor suppressor genes behave in a recessive fashion. Here you can see oncogenes and why they behave in a dominant fashion, right? So here's a homozygous wild type allele set. You know, you've got your homologous chromosomes. They provide normal growth stimulating factors, normal growth factors, normal cell division. But you mutate one allele. You mutate one allele. So this proto-oncogene got mutated to an oncogene. Now the oncogene is making a hyperactive stimulatory factor like that growth receptor I told you about that just tells the cell divide, 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 right? And then this allele, which was not mutated, is only going to tell the cell to divide when it's appropriate, right? But who cares if that one's around if the other guy's telling the cell to divide? You only need one copy of this bad allele, one oncogene, to cause excessive cell proliferation and tumor growth. Now with tumor suppressor genes, on the other hand, tumor suppressor genes, remember these alleles, these yellow alleles, are going to code for normal growth limiting factors. Policemen, they say, hey, don't divide, there's something wrong with the DNA. Hey, don't divide, there's something wrong with the microtubules, right? Or hey, don't divide, there's no growth signal. So you get normal cell division, but then you get mutation in both alleles. So now neither of these alleles is being expressed into a policeman, into a inhibitory protein, right? Does that make sense? The checkpoint is gone. The checkpoint is effectively gone. So now you're going to have excessive proliferation because there's no one to stop the cell at that point to say there's something wrong. Does that make sense? So again, tumor suppressor genes behave in a recessive fashion. Oncogenes behave in a dominant fashion. Here are some examples of oncogenes and the functions, their corresponding proto-oncogenes. So here's an oncogene, okay? This is the mutated oncogene. This is the normal function of the proto-oncogene that it came from. The proto-oncogene was part of growth factor receptor. And so, you need a growth factor receptor. This was the example I was telling you about. So the ERBB gene, normally, its job is to be a receptor, and if there's growth hormone around, to tell the cell divide, right? But you mutate this guy, and it becomes this oncogene, ERBB. Now the oncogene codes for the same receptor, but it's stuck on mode. It just wants to tell the cell to divide regardless of what's going on, regardless of if there's growth hormone around. Does that make sense? So now the cell just keeps wanting to divide because it keeps getting this faulty signal that there's growth hormone around. And there's many examples of this with different oncogenes that have become, mutated, or proto-oncogenes that have become mutated to oncogenes. So again, like I was telling you, the tumor suppressor genes are recessive, so you need two copies. You could be born, for example, heterozygous. So I have one normal tumor suppressor and one mutant tumor suppressor. And if I was born with one mutant tumor suppressor gene, this would be an example of hereditary cancer, right? Because now, just by the nature of being born with one faulty tumor suppressor allele, now guess what? I am at much higher risk of cancer, because at any time in my life, if this other allele becomes damaged, like a deletion happens, now I don't have a tumor suppressor. This cell is effectively recessive, homozygous recessive for allele A, and I could get cancer. Does that make sense? So being born heterozygous for a tumor suppressor gene is a form of hereditary cancer, right? Because now I'm at huge risk, much higher risk of cancer than someone that was born with two big A's, right? Someone that was born with two tumor suppressor genes. Does that make sense? They would have to damage both genes in order to get cancer. I would only have to damage one gene, or one allele, I should say. Okay, so you can see here, these are examples of tumor suppressor genes and their normal function. So the biggest one is P53. You may have heard of P53. Its normal job is to regulate cell division, right? But if you mutate both copies of P53, that's found in many, many different kinds of cancers. And another famous one is BRCA1. This is a tumor suppressor gene that directs DNA repair. It's also a transcription factor. And when you damage both of the BRCA1 genes, that leads to breast and ovarian cancer. That's why they screen for BRCA1 in people who are at risk for breast cancer. Okay, concept check number 2. Why are oncogenes usually dominant in their action, whereas tumor suppressor genes are recessive? We just touched on this so much, didn't we? Oncogenes are dominant because they are coding for, they're mutant proto-oncogenes, first of all, but they code for stimulatory proteins, proteins that stimulate proliferation. So you only need one stimulatory protein to stimulate proliferation, stimulate tumor formation. Whereas tumor suppressor genes, these alleles code for inhibitory factors. Stop proliferating, right? If you delete one allele, there's still the other allele making the inhibitory factor. You need to disrupt both alleles, right? So that's what it says here. All right, a couple more concepts, and then we'll head over to a break time. So, genes, more genes that control the cell cycle. So it's not just these tumor suppressor genes and oncogenes. We also focus on these cyclin-dependent kinases, or CDKs. These are very, very important proteins in the cell cycle called CDKs, which function only when bound to cyclins. So think about it like this. There are the cyclin-dependent kinases. These are proteins that play a role in the cell cycle, but they're only active when these other proteins called cyclins are around. And the cyclins, just like their name suggests, the cyclins are only available at different parts of the cell cycle. So during G1, you might have a lot of one type of cyclin in the cell. During G2, you might have a lot of a different cyclin in the cell. And only when those cyclins go to a high concentration can the cyclins interact with the CDKs, the cyclin-dependent kinases, and activate the cyclin-dependent kinases. When the cyclin-dependent kinases are active, they do their job. Kinases, when they're active, they are proteins that phosphorylate other proteins. Usually, when a kinase phosphorylates another protein, it activates the next protein. This is all part of what's called cellular signaling or signal transduction. This protein gets activated, which then activates the next protein, which then activates the next protein, and then that could lead to cell division or, some kind of cell event, right? Or gene transcription, cell division, etc., right? So again, the levels of cyclin oscillate over the course of the cell cycle. During the G1 to S transition, for example, the retinoblastoma protein plays a big role. During the G2 to M transition, MPF, or the mitosis-promoting factor, plays a big role. During spindle assembly checkpoint, there's another cyclin that plays a big role, right? So you can see that if you mutate any one of these factors, or the cyclin-dependent kinase itself, you mutate that, you could disrupt the cell cycle. Let me just show you real quick how the retinoblastoma gene works. Okay, so here is a gene, okay? You want to activate the gene, but in order to do so, here's how it works. Let's start at the beginning. The retinoblastoma protein binds to another protein called E2F, keeping E2F inactive. Now, at some point, the cyclins, remember the cyclins with cyclin D, which is bound to CDK, and cyclin E, which is bound to CDK, they phosphorylate, their kinases phosphorylate the retinoblastoma protein, inactivating the retinoblastoma protein, which releases E2F. E2F is a transcription factor. It can then travel to the nucleus and bind to a special promoter to activate a gene, right? And this gene is required for DNA replication. This gene produces a gene product which is required for DNA replication, and replication can occur. Does that make sense? So if you were to mutate the retinoblastoma gene, right? You were to mutate the retinoblastoma gene. well, then E2F would not need the signals from cyclin D or cyclin E with their kinases, and E2F would always promote transcription of this DNA replication gene. Does that make sense? So this would be an example of a tumor suppressor. The retinoblastoma would be a tumor suppressor because you'd need to knock out both of the alleles for this guy in order to allow E2F to constantly promote DNA replication. You don't need to know this in great detail, but I just wanted to give you an example of how this might work in cancer, right? How mutating some of these important genes might lead to overstimulation of cell proliferation. Okay, last concept, then we'll take a break time. Do you guys remember telomeres, right? The ends of our chromosomes are called telomeres. The ends of the chromosomes are called telomeres. And do you remember I said every time the cell divides, that causes the chromosomes to shorten a little, and this is called shortening of the telomeric ends. And there's an enzyme that your stem cells and your other highly proliferative cells have that counteracts this. And by other ones, I mean the stem cells and the germ cells have a special weapon against chromosomal shortening, and that's called telomerase. Telomerase is normally active in stem cells and germ cells, but not normally expressed in somatic cells. You know why? Because your somatic cells are not meant to divide all that many times before a stem cell replenishes them. Without telomerase activity, somatic cells can only divide so many times due to chromosome shortening without ceasing cell division and cell death. However, in many tumor cells, many tumors, telomerase is upregulated, so it's ectopically upregulated, allowing cells to replicate indefinitely. So these cells, these somatic cells, can now mutate indefinitely. The cancer cells are said to have become immortalized. Isn't that interesting? Did you guys follow that? Your stem cells and your germ cells are the only ones that are supposed to be able to, replicate indefinitely. Your somatic cells, like your skin cells, they're not supposed to replicate all that much without stalling out. This is by design. However, many cancers have ectopic upregulation of telomerase. That means faulty and out of place upregulation of telomerase. And because of that, those cells are effectively immortal. They can keep growing. And that's one of the hallmarks of cancer, is immortalized cells. Isn't that interesting? In fact, telomerase is a big field of study in aging and cancer. Too much telomerase, cancer. Too little telomerase, aging. Isn't that interesting? It plays a role. It's not responsible for aging and cancer, but it plays a big role in aging and cancer. So concept check number 3, and then we'll take a quick break time. Which type of mutation in telomerase could be associated with cancer cells? Which type of mutation in telomerase? Okay, mutations that produce an inactive form of telomerase can cause cancer? No. I mean, if you don't have telomerase, then the cells can't replenish their chromosomal ends. They can't extend their telomeres, so the cells are not going to become immortalized. Mutations that decrease the expression of telomerase? No. Again, that's not going to help you fix your chromosomal ends and become immortal. Mutations that increase the expression of telomerase? Yes. Now you have too much telomerase. The cells have no problem with shortening of the chromosomes. The cell can divide forever, and that's essentially what cancer is, so it has to be C. Does that make sense? Isn't that interesting, right? So with that, let's take a quick little break time with Gizmo and Wicket, and we'll come back strong to finish this chapter off. What do you say? All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and keep going, finish off this chapter strong. Where we left off, we were discussing the important genes in cancer development, and one type of, one area of cancer that is very important is this area of vascularization. These genes that allow blood vessel growth to occur for the tumor cells. Now, tumor cells, once a tumor forms, it becomes a ball of cells. It becomes exceedingly difficult for the cells in the center of the ball to obtain nutrients, right? Remember what the vasculature does, the blood vessels in your body, what it does. It carries blood. That blood carries with it oxygen. It carries away carbon dioxide. In the blood, you also have nutrients like sugars and amino acids and such that the cells need in order to grow and survive, right? So what's really interesting is that early researchers and doctors that were studying cancer, they would notice that when they would remove tumors from patients, these tumors would be highly vascularized, meaning there were these weird blood vessels growing into the tumors from every angle that normally those blood vessels wouldn't be in that tissue. It's almost as though the tumor itself promoted angiogenesis, angiogenesis means new blood growth, right? Novel blood growth. And that's exactly the case. Angiogenesis is required for tumor growth and survival. These clever tumor cells secrete angiogenic factors. These are proteins, signals that bring new blood vessel growth into the tumor. So angiogenesis is stimulated by growth factors that are secreted by the tumor cells. Tumor cells overexpress these growth factors. For example, VEGF is a vascular endothelial growth factor, VEGF. And so one area of study is producing inhibitors of angiogenesis to assist with limiting tumor growth. Imagine if you give someone a medication and it's essentially a drug that blocks angiogenesis. It prevents new blood vessel growth. Well, that's one way of preventing that patient's tumors from getting bigger, right? Because they won't be able to recruit new blood vessels. The tumors won't be able to grow in size. And that's an interesting avenue. And a lot of times these anti-angiogenic factors, these drugs that prevent angiogenesis, are prescribed along with chemotherapy or radiation and, like a prescription as part of their prescription regimen. What else is there? There's also epigenetic changes associated with cancer. Remember what epigenetics means? Usually it has to do with DNA methylation or chromosome structural arrangement. So you could alter DNA methylation profiles either by having too much methylation or too little methylation. You could alter chromatin structure, and that's also seen. Strange epigenetic changes are found in cancer cells frequently. So here's an example of sequential mutation. Do you remember what sequential mutation means? It's part of the progression of cancer. You get one mutation, and that makes a cell more likely to proliferate, and then another mutation, the cell proliferates too much. A third mutation, now it's proliferating, and it has a bizarre cell shape to it. You have abnormal cells. And then you can have, or irregular cells they call them, and then the fourth or fifth mutation, now you have metastasis. So I'm going to show you a progression model in the next image. Okay, so remember, again, most cancers arise from mutations in several genes, not just one gene. very few cancers are caused by one gene. Often a combination of oncogenes and tumor suppressor genes need to be mutated. Colorectal cancer, this is one I'm going to show you. Colorectal cancers arise in cells lining the colon and rectum as benign tumors called adenomatous polyps, and then they progress into metastasis. And here's how it works. So here, this is part of your colon, and this is a normal colon with normal colon cells. And then let's say, and this is a cross section of the colon, obviously. Let's say you lose an important tumor suppressor gene called APC. Okay, so now a small polyp will form, a tiny little polyp. Small growth forms on the colon wall. And this is called a polyp, and it's just benign. It's sitting there. It's a cell that's incurred one mutation. It's not going to be full-fledged cancer. All right, it's just a little polyp, just a little bit of proliferation going on. So a benign precancerous tumor grows. And here you go. Here's your little precancerous ball of cells with a single mutation in the tumor suppressor gene APC. Now, you get a second insult, right? That you have activation of oncogene RAS. So somehow the proto-oncogene becomes mutated or misregulated and becomes an oncogene RAS. So now you have an oncogene present as well. Remember, oncogenes are stimulatory signals. So now not only are you missing a APC tumor suppressor, but you have a stimulator, this oncogene RAS, so the tumor becomes bigger, right? An adenoma or benign tumor grows. Okay. Next, you lose another tumor suppressor gene. Remember we discussed this important tumor suppressor called P53? You lose another tumor suppressor, and now it grows even more. A carcinoma forms, which is a malignant tumor develops. So malignant, look what it's doing. It's starting to invade adjacent tissues. Malignant tumors invade adjacent tissues. Next, you lose an anti-metastasis gene. And I'll explain this more in a little bit. And basically that's another insult. And you have a cancer that metastasizes, which means spreads to other tissues through the bloodstream. Okay, so let me show you something here. This is from a very interesting and important review called The Hallmarks of Cancer. And I want to tell you the authors because I want to give them credit. No, that's not it. Where are the authors are Hanahan and Weinberg in 2000. They published a very interesting review called The Hallmarks of Cancer. And in it, they said that cancer cells are able to do these things. You know, once they're, metastatic, once they've gone full-fledged cancer. And that is, cancer cells sustain proliferative signaling, so they have constant signal to proliferate. Remember, oncogenes? They evade growth suppressors, so tumor suppressors don't work on them. The tumor suppressors say, they don't work anymore. They activate invasion and metastasis genes, right? They are invading, and they are not just invading other cells, but they can spread to other cells. They can replicate forever, right? They're immortal. Remember, that's where telomerase comes in. They're immortalized, right, with the help of, for example, telomerase. Now the cells can divide forever. They induce angiogenesis, remember, new blood vessel growth. So things like VEGF, this vascular endothelial growth factor, right, causing new blood vessels to form, right? So now they have an endless supply of nutrients to grow. And they resist death. So they don't respond to normal apoptotic signaling. They don't incur cell death, program cell death either. Once a cell has incurred these insults and lost this kind of control, this is full-fledged cancer, and it's very aggressive and it spreads everywhere in the body. Another thing is, let me explain a little bit more about activating invasion and metastasis. I just want to explain a little bit to you. A normal healthy cell has what's called anchorage dependent growth. Okay, a normal healthy cell requires anchorage for growth. What does that mean? A skin cell, for instance, is a happy, healthy skin cell. It needs to be attached on each side. It needs to be anchored to other cells in order to grow. If you were to take a skin cell and detach it from all neighbors, that means there's no anchorage, right? It's not attached to neighbor cells. Most cells, healthy cells, cannot grow in that situation. They know something is wrong. Like if I'm not attached to my neighbors, I shouldn't be growing. Does that make sense? However, metastatic cancer cells have incurred mutations that not just allow them to invade, but to undergo anchorage-independent growth, right? Anchorage-independent growth. That means that they will continue to divide even if they're not attached to other cells or any other tissue. Does that make sense? That's another hallmark of cancer. Interesting. So let's get back to this. Again, chromosomal instability is a general feature of cancer cells as well. A lot of cancer cells exhibit deletions, inversions, translocations. You could even see duplications. You can see aneuploidy. Aneuploidy is very frequent in cancer cells. Example, a reciprocal translocation between chromosome 9 and 22 causes chronic myelogenous leukemia, right? So you could get a form of leukemia just from a translocation. So here's an example. You've got the ABL gene and the BCR gene, and they translocate. This is called the Philadelphia chromosome. And this is a cause of chronic myelogenous leukemia. So this form of leukemia is caused by a translocation event. Again, that's another insult to the chromosomes. That's a mutation. That's a type of mutation. It's a chromosomal mutation, but it's a mutation nonetheless. Chronic myelogenous leukemia is usually associated with which type of chromosome rearrangement? Wasn't it a translocation? A translocation. And look here. Remember I said cancer cells are often aneuploid? Cancer cells often possess chromosome abnormalities, including extra chromosomes, missing chromosomes, and chromosome rearrangements. So take a look at this. This is from a cancer cell. This is from a colon cancer cell from some patient. It looks like they're missing a chromosome 1. Chromosome 3, something's wrong with it because it looks like part of it's missing, so it probably translocated a tip. Chromosome 5's, one of the chromosome 5 homologs is missing, and then there's, what in the world is going on here? Oh, here's that part of 3. Look, here's a fragment of 3. There's where it went. And then there's a part of 12. Part of 12 is over here. So these chromosomes have actually fragmented somehow. You're missing 8, you're missing a 9, you're missing a 10, you have too many 11s. So do you see, long story short, aneuploidy is very common in mutated cancerous cells in cancers. And like I said earlier, viruses can give you cancer. Viruses are responsible for a number of cancers in animals and in people. Retroviruses, for instance, cause cancer by mutating and rearranging your proto-oncogenes. Remember, proto-oncogenes are your normal genes that when misregulated or mutated can cause cancer, right? What What if a retrovirus infected you and it inserted a strong promoter next to one of your proto-oncogenes? Think about it. Now you're getting too much expression of that proto-oncogene, and that might actually stimulate too much growth, and that might cause that cell to divide too frequently. So here's an example. Human papillomavirus, or called HPV, there are certain strains of HPV that cause cervical cancer. In fact, 95% of all women with cervical cancer are infected with those strains of HPV. So this virus gets in, it affects your normal cell cycle regulation by affecting cell cycle genes, and those cervical cells that it infected replicate too often, right? Here's a list of viruses and the human cancers that they cause. So human papillomavirus, cervical cancer, hepatitis B virus, liver cancer, Epstein-Barr virus. This is the virus called mono. If you've heard of high school kids, for instance, getting mono. Epstein-Barr virus caused Burkitt's lymphoma, and so on and so forth. So it's not uncommon for viruses to cause cancer, and the way they do that is either by disrupting tumor-suppressor genes or proto-oncogenes, upregulating proto-oncogenes, putting promoters in front of proto-oncogenes, causing deletions, causing DNA damage, you name it. What they're doing, though, is they're affecting your cell cycle genes in some fashion, and that's giving those cells they're losing their ability to regulate their cell cycle, and they're dividing too much. All right, so let me give you a couple examples of how retroviruses can cause cancer. First, I'll show you how a retrovirus can cause cancer by mutating and rearranging proto-oncogenes. And next, I'll show you how it can cause cancer by putting a promoter down in front of a proto-oncogene. So let me show you the first type. Here's your retrovirus. It's infecting your cell. Okay, it infects your cell with RNA. The RNA is reverse transcribed to DNA. That DNA then inserts itself into your host chromosome, into your chromosome. So now you have the viral genome next to a proto-oncogene. Then when the virus reproduces, it could actually package not just its own genome, but it might package the proto-oncogene as well. Does that make sense? So now the virus has not only packaged its own genome into new virion, but it has also packaged your proto-oncogene into that virus as well. So when that virus infects the next cell, it's going to introduce its own genome as well as the proto-oncogene. And when the viral genome and the proto-oncogene recombine into your host genome again, this produces an oncogene that is inserted back into the host chromosome, and that causes cancer. Because now, because now that proto-oncogene is known as an oncogene because it has become mutated into an oncogene. So do you see what happened? In the first round of infection, the virus was able to insert its genome next to your proto-oncogene, and then it was able to remove that viral genome along with the proto-oncogene, package it into virus. Sorry, I need to take a drink here. Sorry about that. So then the virus was able to package that proto-oncogene into new virion, and when it infected the next round of cells, it not only introduced its own viral genome, it introduced that proto-oncogene, and that proto-oncogene may have formed into an oncogene through mutation, and now it's an oncogene. So now you've got too much stimulatory signal in this new host cell, and that could cause that new host cell to divide too much because it's getting too much signaling, because it has either two copies of this proto-oncogene, or in this case it's got mutated proto-oncogene to a full-fledged oncogene. You see what I'm saying? Either way, it's because of the virus moving about and causing mutation in your proto-oncogene that your cells now have too much signal. Here you can see another effect of viruses. So here your virus infects your cell with the RNA. It gets reverse transcribed to DNA. In this case, look what it does. It inserts the provirus next to a proto-oncogene, and it has a strong promoter. It has a strong viral promoter, and that causes upregulation of the proto-oncogene. So you have overexpression of the proto-oncogene. If you have too much of this proto-oncogene, it might give too much stimulatory signal and tell the cell to divide too frequently, and that could lead to cancer. All right, so I hope this was interesting. It sure was for me. I love talking about cancer genetics because, again, it has everything to do with genetics. If you have a full understanding of genetics, you do have a very good grasp on cancer. And now, hopefully, you have an appreciation for why it's so difficult to study cancer and why, they say, well, people have studied cancer for how many years now and there's still no cure. I hope you can get an appreciation for why there is no cure, because it's a heterogeneous disease. It's not caused by one thing. No two cancers are caused by the same thing. You have different tumor suppressor genes that could be disrupted. You have different oncogenes that could be disrupted, or proto-oncogenes that could be disrupted. You have different cyclin-dependent kinases or cyclins. You have telomerase going on. You have angiogenic factors. You have viruses getting in there and causing issues. You have smog and cigarettes and alcohol and weird chemicals in there. And you've got the sun and you've got x-rays, and all of these things are causing different problems in different cells with different issues. And so no two cancers are really the same. And also the big factors are when you catch the cancer, when do you spot it, when do you start treatment. It's such a complex area. It's not like getting a bacterial infection. If you get a bacterial infection, you can take a pill that kills bacteria. But if the problem is your own cells dividing too much, you can't take a pill that kills your own cells, because that pill will most likely kill you. Does that make sense? So it's a very, very complex problem. It's, for every, time we make strides, we realize how many strides there are still left to take to solve this problem. And hopefully now you have an appreciation for why and a greater understanding of how cancer works. So with that, if you have any questions, leave them in the comment box below. Otherwise, I will catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D,